Chapter 3 introduces us to the basic molecules inside the body. We know what the four types of molecules are. They are carbs, better known as sugars, lipids, also known as fat, proteins, and then we have nucleic acids. It's this last one that's probably the least familiar, but examples of nucleic acids would be DNA and RNA. The molecules that are responsible for why we look like our parents, we resemble them in some way, because that's what's inherited. Now, before we go further, there's something here that I want to show you that I think is very important. If you take a look at these two molecules, you have estrogen on the left-hand side and testosterone on the right-hand side. What do you notice? How many rings does estrogen have? Well, estrogen has 4 rings. How many does testosterone have? It also has 4 rings. Both of them, estrogen has an OH group on the bottom left-hand side, and it has you'll see a double bond with an O. That's really a C double bonded to an O. The pointed edges of the ring are assumed to be carbon atoms. Anyway, the C double bonded to an O, that's the carbonyl group. So estrogen has 2 key parts, the OH down at the bottom left-hand side and the C double bonded to an O on the upper right-hand side. Those are the 2 key parts of estrogen. The rest of the molecule is not as important. You'll notice that testosterone also has 4 rings, and it has the same 2 functional groups, but the difference being that the OH is now on the upper right-hand side, and the C double bonded to an O is on the lower left-hand side. And yet when we think of these 2 molecules, we associate them as being diametrically opposed. Estrogen obviously we associate with female characteristics, female reproductive organs, as well as traits, feminine qualities, if you will. And testosterone we associate with, again, manly qualities, right? Male sex organs, maybe aggressiveness, things that we normally associate with masculinity. But on a smaller scale, when we look at them biologically, they're actually very similar. The difference is really the location of the 2 functional groups. And basically these functional groups, they are, if you had to define them, how would you define them? Well, functional groups are basically small parts of a larger molecule that tend to be very reactive. So they're the key pieces of a larger puzzle. So here I define functional groups as the most reactive part of a molecule. 6 functional groups that I absolutely want you to know. The hydroxyl group, which is simply an OH, and you saw that both in estrogen and testosterone. The carbonyl group, C double bonded to an O. The carboxyl group, which is C double bonded to an oxygen and then an OH group, or COOH. An amino group, which is an NH3, a methyl, CH3, and then a phosphate group, which is a PO4, a phosphorus attached to 4 oxygen atoms and has a charge of -3. Don't worry about the charges, but these 6 groups you will see over and over again. These are just different models used to represent specific molecules. So in this particular case, glucose is drawn. In the first instance, you can see what's referred to as the ball and stick model for glucose. And this is just a way to show us what glucose looks like. So the balls that are in black, those are the carbon atoms. And then, you have the red balls, which are the oxygen atoms, and then the white balls, which are the hydrogen atoms. Here's part B. We can see, again, ball stick model for glucose, just a different angle. In part C, glucose is represented using a ring structure. And then D, the same idea. But there are different ways to represent glucose, and this is just my way of introducing those different ways to you. So if we are building larger molecules inside the body, we have to start somewhere. And so one of those ways is by having, building blocks, right? Just like you'd use bricks to make a house, you need building blocks to make larger items inside the body. So we know simple sugars can be used to form larger carbohydrates. Fatty acids are used to make lipids or fat. Amino acids are used to make proteins. So if you take, 2 or more amino acids and you link them together, they form proteins. And then nucleotides form nucleic acids. So if you've seen a long DNA chain, that's basically nucleic acid, and that's formed by tying up a bunch of DNA letters together. And the way we do this is through a process called condensation. Condensation takes smaller pieces and uses them to make one larger piece, right? Smaller pieces, in this case, the biological term would be monomers, and the larger piece here, polymers. Polymers being many, monomers, mono meaning one. Here you can see the two different types of reactions that I want you to be familiar with. And they're both fairly intuitive. You can see in condensation what's happening. You have two small sugar molecules being combined to form a larger sugar molecule. And the process where they release water, which is represented by the HOH. Of course, the reverse of this is hydrolysis, where a larger molecule has a bond between it, and that bond is broken, and what you get as a result are two smaller molecules. So condensation is a process in which an enzyme takes two small molecules and puts them together, combines them into a larger one. Whereas hydrolysis is a process in which the enzyme splits a larger molecule into two smaller ones. So these are basically the reverse of one another. The three main types of carbohydrates that I want you to be familiar with: monosaccharides, oligosaccharides, polysaccharides. Again, mono meaning one, oligo meaning a few, and poly meaning many. Monosaccharides are also known as simple sugars. Oligosaccharides are basically sugars that are small chains that have a few sugars or a handful. But if it's a carbohydrate that has hundreds of sugar units attached together, that's called a polysaccharide, which are better known as complex carbs. So then, what is the role of carbohydrates in the body? And more specifically, why do we need carbs if we also have fat and proteins that can serve as sources of energy? Meaning that if our body needs energy, we can get the energy from fat, we can get it from protein. So what makes carbs so special? And what makes carbohydrates so special is that they provide energy faster. So pound for pound, fat stores twice as much energy as carb, but it's not as quick. So your body will, extract energy from fat, but if you have a race to run in a short time, you need energy now and not tomorrow, and therefore you may be better served by eating carbohydrates. Of course, energy is not the only thing that is extracted from carbohydrates. Carbs are broken down inside the body, and the broken pieces are recycled and used to build structures inside the body. So carbohydrates are generally made up of carbon, hydrogen, and oxygen. Well, the carbon, the hydrogen, and the oxygen are recycled to build tissues and organs, to regenerate cells inside our body. Here we can see sucrose, which is a disaccharide. It's formed from two monosaccharides, right? So what type of reaction would this be? This would be condensation. When you think condensation, think combining things. Lipids, as we said earlier, are a source of fat, right? Lipids is just a fancy word for fat. And they also provide energy, just as carbs do, but they do so more efficiently. And specifically, as I said to you, pound for pound, or gram per gram, if I give you a gram of fat, and I give you a gram of carbs, the gram of fat has more than twice as much energy, right? Now, these lipids, or fat, are also used to build structures inside the body. Specifically, one key area is to make the cell membranes. There are other functions as well. We saw earlier testosterone and estrogen, which are basically lipids. Triglycerides represent one way that our body stores energy as fat. And so each triglyceride contains 3 fatty acids. Here's a picture. This figure shows what triglyceride looks like. And what it has is, you can see over here, there's a glycerol molecule. Don't worry about that name. You don't have to memorize it. It's attached to 3 finger-like structures, these projections, which are the fatty acids. The fatty acids, as you can see, are mainly carbon atoms with hydrogen all around them. Each carbon can form up to 4 bonds. You see 4 lines around each carbon. Those are bonds. So we have 3 fatty acids attached to a glycerol molecule, and that's what a triglyceride is, right? Tri meaning 3. Penguins use triglyceride as a way to store energy in their adipose tissue, so that if they're going through a long winter and there are extended periods where they're having trouble finding food, they have this stored energy as a reservoir, as an energy reservoir. Phospholipids are a type of fat. Specifically, phospholipids, don't worry too much about the structure over here. There's a hydrophilic part and a hydrophobic part. We will get to that later on in chapter 4. But for now, all I really want you to know is that phospholipids are a type of lipids that are used in the cell membrane. Okay, so when I said that lipids are important for us because they help to form the cell membrane, which is the outer layer of the cell, which keeps the inside content of the cell on the inside from flowing out. Proteins are very, very diverse. So carbs, we said, provide energy and help with the structure. Lipids also help in terms of energy and structure. Proteins have so many different roles. We can start by talking about how they help to form the body structures. An example would be collagen, which is a protein in the skin that helps to keep the skin firm. Proteins are also used as enzymes. And enzymes are, they help speed up reactions inside the body. So whether it's breaking things or building things, they make everything happen faster. And so pepsin would be one example of an enzyme, or one example of a protein, I should say, that help to break down other proteins inside the stomach. So when you're digesting food, pepsin is helping to do that. Proteins can also help communicate inside the body. How does the brain communicate with the kidney or the liver or other remote parts of the body, it does so through the use of proteins. One of those proteins being, fancy word that you'll hear, are receptors. Receptors are proteins that are on cells that pick up signals from the outside world, either through hormones or neurotransmitters, which we'll talk about later on. Proteins also help to defend the body against pathogens, such as bacteria and viruses. So when you hear the term antibodies, what are antibodies? Antibodies are not cells. They are proteins that bind to bacteria, viruses, and other foreign pathogens. There are 20 different amino acids in our bodies. It is these 20 amino acids that are used to make proteins in different combinations. So just like we have, in the English language, we have 26 different letters, and those letters are used to form words, right? We don't use each letter, all the letters equally. Some letters are used more often than others. For example, the letter A, B, or T is used far more often than, say, the letter Z. Okay, but yet we do have 26 letters. Same way, there are 20 different kinds of amino acids. Not every single amino acid is used. They're not all used equally, but they, some are used more than others. But these are the amino acids that are used to make proteins, and they all have a general structure like the one you see here. There's a carbon in the middle. There's an amino group on one side, a carboxyl group on the other side, and a hydrogen at the top. And then it's a bottom portion that's different. So for valine, that green box that you see, that's what contains the pieces of valine that makes it different from all the other amino acids. I don't expect you to memorize the structures for the amino acids, but you should know that there are 20 different kinds of amino acids. Here's an example of two amino acids being combined into a protein, a process called protein synthesis. So MET stands for methionine, and it's being combined with ALA for alanine, and together they are forming what's called a dipeptide, di meaning two, peptide meaning protein. So you'll see the term protein or polypeptide being used. In this diagram, so MET and ALA are being combined into a dipeptide, and then a LEU, which is a third amino acid, is being combined with the first two to form a larger one. And so the process continues, this process of protein synthesis. There will be proteins that are 20 amino acids in length, or 100 amino acids in length, or a couple hundred. It can really vary. But the idea being that two or more amino acids are used to make the protein. Secondary structure, tertiary structure, and quaternary structure. The primary structure simply refers to how those amino acids are arranged, meaning in what order. And so if you take the amino acids and you lay them flat as you would beads on a string, the order in which those amino acids appear, that's called the primary structure. The secondary, the tertiary structure refer to what happens to, if you will, the beads on a string once they are twisted and turned. What shape do they take? The shape of protein greatly determines what type of job it will do, how effectively it will do the job. And I'll give you an example of that. The key point here, though, being that the shape of a protein, whether it's round and fluffy, or whether it's rod-shaped, or whatever else it might be, greatly impacts what it does, what the protein does. Here we see the primary structure of a protein, a bunch of proteins linked together. This is the first amino acid on the left-hand side, and each box contains one amino acid. So there's 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 amino acids linked together in a polypeptide chain. And remember, as I said, a polypeptide is a fancy word for protein. So, protein structure dictates function. If the protein does not have the right shape, it can't do its job. A good example of this is the role that hemoglobin plays in sickle cell anemia. Hemoglobin, as you probably know, is involved in the transport of oxygen from the lungs to the different parts of the body. Here you see a structure of hemoglobin. There's a ribbon there's really 4 ribbons here, okay? 4 ribbons, and each ribbon carries an oxygen molecule, an O2. That red little plate that you see within each ribbon, there's 4 of those red plates. That's where the oxygen is held. So it's basically like the seat where an oxygen molecule is held still. The important thing about these ribbons is that they're not just randomly, twisted and turned. They're done so in a very specific manner. And if you change the shape of those ribbons, if you clip a piece off, or if you twist and turn it more than it naturally twists and turns, then the oxygen molecules won't fit in. And what happens is that in the case of sickle cell mutation, so sickle cell mutation, here we can see there's a bunch of amino acids that are connected together into a polypeptide chain. So these amino acids will combine together to form the polypeptide that gives hemoglobin its shape. So as I said, hemoglobin is a protein. It's made up of a bunch of amino acids. In sickle cell mutation, one of the amino acids is incorrectly substituted. So where there should be a glutamate, there is valine, which means that the shape of the entire hemoglobin changes. And hemoglobin is unable to carry oxygen as it normally would. Hemoglobin is found in red blood cells, in the bloodstream, and when hemoglobin shape changes, the shape of the red blood cells change. So instead of being round and fluffy, those red blood cells look sickle-shaped. They're hard, and they're no longer round and fluffy. And because they're hardened, these red blood cells have a harder time going through the bloodstream. Bloodstreams are basically narrow channels, that have limited space for the cells to move around. And if these cells become hardened, then it creates friction against the blood vessel walls and causes these cells to break down. What does that result in? All sorts of problems. These red blood cells can't do their jobs. There's circulatory problems. Oxygen's not getting to the brains. To the brain, to the heart, to the lungs, to the gut, the kidney, and results in systemic problems throughout the body. These red blood cells also then need to be, because they're damaged more frequently, they need to be recycled more often, and the place they're recycled in is the spleen. So the spleen has to work harder. And that can result in things like anemia, which obviously causes weakness and fatigue, and that can affect a person's development. So, when a protein loses its original shape, its 3D shape, it is said to be denatured. That denaturation, or the loss of shape, can result from a change in pH, as pH is something we talked about in Chapter 2, or temperature, like in the case of, say, a fever. During a course of a fever, what happens? A person's temperature rises, and the proteins inside their body are not able to function like the way they normally would. So pH is a cause of denaturation, temperature change, as well as exposure to detergents and salts. Because all of these factors, they help to disrupt the hydrogen bonds that help to hold a protein's shape in place. The last macromolecule that we need to talk about are nucleotides, which are the building blocks of DNA. A nucleotide consists of a sugar, a phosphate group, and a nitrogen-containing base. So in this figure, the double ring in blue, that's the nitrogen-containing base. The five-sided ring in orange, that's the sugar. And then the three phosphates are on the left-hand side. These are the four different nucleotides of DNA. So we have adenine, thymine, guanine, and cytosine. And overall, they have a similar structure. Each of them has a sugar in the middle and a phosphate group on the left-hand side. And what they really differ in is the base that they carry. So in the case of adenine and guanine, the bases are double-ringed, whereas in the case of thymine and cytosine, the bases are single-ringed. Adenine is symbolized by the letter A, thymine by the letter T, guanine by G, and cytosine by C. You want to make sure you know these four types of nucleotides because they are the letters that are used to form the DNA words. So just like the English language has 26 letters, the DNA language has only 4 letters, and it is these 4 letters that are used to form the DNA words. So DNA is double-stranded. I'll explain what that is in a minute. It contains 4 types of bases, as we just discussed, and DNA is the permanent copy. There's another type of nucleic acid, RNA, which is not the permanent copy, it's actually the temporary copy. Now here's an important piece. Adenine pairs with thymine, guanine pairs with cytosine. So if I give you a template strand, meaning if I tell you that your original letters on one side are A, G, T, T, A, C, T, and I ask you, what's the complementary strand, right? As I said, DNA is double-stranded. So if I ask you, well, what's the complementary strand? You would say, okay, well, we know A pairs with T, so A would go with T, G would go with C, T would go with A, the second T with A, the A with T, the C with G, and then the T finally with an A. And so that would be your complementary strand. RNA is usually single-stranded. It also has 4 types of bases. The first 3 are the same. It's the 4th one that's different. So instead of having thymine, RNA has uracil. RNA is the temporary copy. So DNA is typically used to make RNA, and RNA is used to make proteins. Transcription is when DNA is used to make the RNA, and this usually happens in the nucleus. The RNA is then used to make proteins outside of the nucleus in what is called the cytoplasm, and specifically in the cytoplasm at a site called the ribosomes. This is something we can discuss more in class, and we'll talk about in Chapter 4. So if I say to you, what is the DNA template strand if the complementary mRNA strand is UAGC, UUC? Well, we know that U pairs with, well, uracil is taking the place of thymine, so it would be U pairs with A pairs with T, G with C, C with G, U with A, U with A, and then C with G. This is something we can talk more about in class. Here's the DNA double helix, and you can see there are 2 sides. So this is what I meant when I said to you DNA is double-stranded.